The title of this presentation is Reverse Translating Amino Acid Sequences. To begin with the presentation overview, we'll be introducing reverse translation of the amino acid sequences. And to perform this reverse translation between a DNA sequence and an input amino acid sequence, we'll have to describe the process of performing the procedure of reverse translation, which we're going to break down from an open source implementation that's offered from Gene Corner, and how one can also make use of the intermediate file steps and intermediate procedures that's offered by Gene Corner for being able to look at reverse translation in different host organisms rather than the reverse translation templates that are provided for a couple of publicly available host organisms, which I'll list out and describe further. And in particular, this reverse translation step includes the steps of inputting some amino acid sequence, choosing the host expression organism, which for this case we'll take to be E. coli, but the reverse translation step can also be implemented and done for other hosts, from which we'll obtain a probability distribution, which is dependent upon the codon frequencies from the sequence expression suite, which I'm also going to provide an overview of, which finally allows us to output some possible desired sequence for the translation. So we're going to be breaking down each one of these steps and demonstrating how the results are dependent upon the expression host organism which someone chooses, as well as the input sequence in a couple of intermediate steps. So we're going to be looking at the sequence manipulation suite, which is a public resource offered to us by Gene Corner, in which we can input the amino acid sequence or even a FASTA file containing however many amino acid sequence which one would like to reverse translate to a DNA sequence. And below, rather than the amino acid sample sequence that we're going to put in for the translation to execute the reverse translation procedure, we can also look at the codon table, which is specifying from an amino acid the codon and the fraction of the codons that appear corresponding to each amino acid sequence, which informs the reverse translation step and the computation of the graph-based probabilities, which we're going to provide, because it determines how frequent different codons are depending upon the input amino acid sequence and how likely it is or how probable it is to obtain a certain desired output sequence from the reverse translation. And this is to say that the step which is involving the codon frequencies, which is given as some input parameters rather than the amino acid sequence at the beginning from the previous slide in the Gene Corner suite, the sequence manipulation suite, is dependent upon codon frequency tables, such as the one that's shown on the slide. We can choose different type of expression host organisms, for example, for E. coli, and we can look at triplets, which are given in the leftmost column of the codon frequency table, which correspond to some amino acid. And in the third column of the codon frequency table for E. coli, we can see that there are certain decimal points or numbers, which is giving the fraction, or which is proportional also to the frequency per thousand, which is given in the fourth column of the table. And it's important to keep in mind the fraction because it's relating to how likely it is for an amino acid to appear after it's being reverse translated from the input amino acid sequence in a certain expression or host organism that we would like to reverse translate to. So this is important because if we have in mind different types of amino acid sequence that we would like to translate, as well as the expression host organism that we want to look at, the content or the structure of the amino acid sequence can differ widely depending upon the host organism that we would like to reverse translate into. And this is just to say that, as we've been mentioning, the codon frequencies can be very different amongst different host organisms. Rather than the codon frequency table that I demonstrated to you for the E. coli host organism, there can also be many other types of host organisms, from human, mouse, rat, pig, or even C. elegans or insects or different strains of yeast or even streptomyces. In the case of streptomyces, we can see that the codon frequency table is given by a similar form, in which in the leftmost hand column of the codon frequency table, we have different possible triplets. But the thing is that the fraction and also the frequency per thousand, which are respectively given in the third and fourth columns of the codon frequency table, can widely differ. And this can impact the reverse translation step that we would like to use when we're computing the graph-based probabilities, which I'll demonstrate to you how these computations are carried out from a sample amino acid sequence, and how this will inform the final consensus sequence that's obtained for the reverse translation. And this is just to underscore the fact that we can see, even from some entries of the codon frequency table for streptomyces on the left-hand side and comparing it to the codon frequency table for E. coli on the right-hand side, that given the same triplets which appear in the first 4 entries of the triplet table, which is in the leftmost hand column of both of the tables, that the amino acid frequency, or the fraction, it can either be 0.02, which is given in the entry in the first column, or 0.58 for the fraction entry that's given for the E. coli codon frequency table. So this is just to demonstrate that the reverse translation sequence or procedure, as we've been mentioning, in which we'll highlight some more of the steps that's involved in this from the input amino acid sequence, can differ a bit depending upon the expression in the host organism that we would like to reverse translate in. And we'll demonstrate the results from the sequence manipulation suite, which can also yield some additional prospects for reverse translating in other host organisms, rather than those which are publicly available from the codon frequency tables from the drop-down menu that we gave, which can be used in the Gene Corner expression suite. So at the very beginning, or the top of the sequence manipulation suite, we can see that there are 2 examples for the reverse translation results. So these 2 results represent the result, or the culmination of the consensus sequence, which the algorithm predicts would be reverse translated in E. coli from the input sequence that we gave. In particular, we say that there are 306 residues for both of the reverse translation sequences. But besides the reverse translated sequences from the amino acid input sequence that are provided at the very top of the sequence manipulation suite, if we go down a little bit further in the sequence manipulation suite, we continue to other results, which include the graph of the base probabilities, which is relating on a computation of a probability distribution, which is looking at the probability of obtaining different type of DNA base pairs in the sequence that we're reverse translating in from the input amino acid sequence. So this is just basically bridging the mapping problem between the amino acids on one side here and the DNA sequence on one side here, in which, depending upon the codon frequency table and how likely it is, or the probability of obtaining a certain DNA base pair, depending upon the fraction of all base pairs of A, C, T, or G in a certain expression host organism, the reverse translation for this host organism, or even for other host organisms that aren't that can also be reverse translated, which aren't from the host organisms which were shown in the drop-down menu, can also be computed. And it essentially boils down to a probabilistic computation problem of determining how likely it is to obtain a certain DNA base pair from the amino acid base pair. And we can continue to other results in the sequence manipulation suite, in which at the very bottom of the sequence manipulation suite, we can see that it has to carry a little more than 300 of these probabilistic computations for determining all possible combinations of the base pairs of the DNA from the input amino acid sequence for being able to reach one of the 2 results for the reverse translated sequence in E. coli. In conclusions that we would like to provide is that we introduced reverse translation of amino acid sequence. We ran the sequence manipulation suite on one short sequence under an expression host for E. coli to determine the reverse translated DNA sequence, which consists of computing graph-based probabilities for different base pairs occurring depending upon the amino acid sequence that we use as input, reaching a consensus sequence for the input amino acid sequence, and finally being able to distinguish between the 2 possible DNA sequence outputs from the computation of these graph-based probabilities. And finally, we conclude that it's possible to adapt these reverse translation algorithms for other host organisms, as we've been mentioning, rather than the ones that I demonstrated in the drop-down menu, which are publicly available from the codon frequency tables. And thanks for listening, and that's it.